So good morning and thanks very much. Part of my job is to stand up here and say, yes, it works, as an investigator. But mostly what I'm going to talk about is how it works and sort of implementing through a specific clinical trial that we have going, several clinical trials. So, conflict of interest statement first. Mayo has licensed a number of the processes that we'll be presenting, and one of the clinical trials that I'll be talking about is supported by, will be supported by a company as such. So, because I'm going to talk about a specific example, and most are not neuroscientists, so I'll give a little bit of a rationale for why we should use adult stem cells in neurodegenerative diseases, and then expand on the role of the GMP facility in moving the clinician investigator into the clinic. So I'm a neurologist, I'm a neuroscientist, I'm not a regulatorily trained person. All of my training is in basic neurobiology and clinical neurology. So to have the ability to work with a facility like the Human Cell Therapy Lab that can get you over these regulatory hurdles is fantastic. And then I'll talk a little bit about the ongoing clinical trials within this. So first of all, the rationale for using stem cells in CNS degenerative disease. First of all, for all of these that I'm talking about, we're not talking about stem cells becoming new nerve cells. Rather, what we're talking about is can they support or protect against specific disease processes. And I'm going to talk specifically about ALS, but what I'm talking would be equally applicable in Alzheimer's disease or Parkinson's disease. And the features of each of these diseases is that a very selective population of nerve cells dies prematurely in the nervous system. One of the major obstacles to treatment is that in all of these diseases, anything that you put into the bloodstream or anything that you take right now is unlikely to get into the nervous system because of the blood-brain barrier. So the blood-brain barrier protects the nervous system, the brain and the spinal cord are floating in the spinal fluid, and drugs that you put into the circulation, especially biologicals, are never going to get to see the target neurons. So the whole rationale here is, can we use, in this case, MSCs as delivery vehicles to cross the barrier? So I'll just say a few words about Lou Gehrig's disease. Some of you may be familiar with it. It's named for who was the #1 batsman in baseball history, according to Time magazine, in 1936. In 1938, his batting average started to drop, and that season he thought it was just because he was 35 at that time. The next year, he could tell there was something, over the winter, he could tell there was something seriously wrong, and he actually came to Mayo Clinic and was diagnosed with amyotrophic lateral sclerosis. And interestingly, in 1939, he actually took part in a clinical trial. This was the era when vitamins were being discovered. There were clinical trials going on at Mayo of a variety of newly discovered vitamins. He gave that famous speech in 1939 where he went to Yankee Stadium and said that he was the luckiest man alive. And then at the age of 38, in 1941, he died. Very typical history, 2 to 3 years from the onset of first symptoms to death. Now, the reason that people with ALS die is because they selectively lose motor neurons. There are two types. In my brain, when I say I want to move my finger, that movement's initiated by upper motor neurons in my cerebral cortex, travels down into the brainstem and the spinal cord to the lower motor neuron, or the nerve cell, that will connect to the muscle and tell it to work. It's those upper and lower motor neurons selectively that are lost in patients with ALS. Now, what we're trying to do then is to develop a platform that will provide protective strategies to those neurons. The reason most people die is because of respiratory failure, because the muscles that control your breathing So there's the summary of the disease, and I won't read through that except to say why this is an ideal neurodegenerative disease to use as a clinical study pilot or a model of how to do this. And it's really down at the bottom here. First is it's relatively rapidly progressive. So 2 to 3 years you go from being normal to death, and there's no effective therapy. So we're dealing with something that's very serious. It's also relatively easy to measure change, and over time the FDA have accepted death or ventilator dependence, a summated muscle score that you can measure of muscle strength, or something called the ALS functional rating scale. So you have things that you can measure the progression of the disease quite reproducibly. And so clinical trials are relatively straightforward to do in a timely way, compared with something like Alzheimer's disease or Parkinson's disease, where you're looking at 10 or 5 to 10 to 20 year to measure progression. Now, as you've seen from that folders that Al showed, this process from getting from bench to bedside is incredibly complicated. And it was at some point in the top half of this complicated diagram that we sat down and started to work with the Human Cell Therapy Lab and through them with the FDA to simplify this and say, okay, we want to move forward in this way. How can we get through this in the fastest way? And one of the things that we did was actually to take out a lot of the steps that have traditionally been used in bringing something from the clinic to, from the lab to the clinic. So we worked with the FDA to say, here's our rationale for doing this. What is the minimum in terms of being able to move this stem cell therapy into patients? And what they said was, well, what you need to do is a safety study. And that safety study is one that we can understand, that we will believe that it is safe to bring these cells into patients. So what they guided us with was the animal model to choose. They said you need to use an animal model, but it's one where you can actually do the procedures that you're planning to do on patients, which involve CSF injection. And in this case, the smallest animal, and hence the cheapest, that you could practically do this in was a rabbit. Choice of cell type discussed, and then what the studies would be that we had to do and how we had to do them in order to be able to submit data to the FDA, which is not the standard way we do business in the lab. So in fact, we ended up doing a rabbit study. The details are set out there. And one of the key points that happened that really cut this process down in time was we said we wanted to do a dose escalation study in patients. And therefore we wanted to do a dose escalation study in rabbits. And the FDA came back and said, no, go to your highest dose first, because if that's safe, then you don't need to do a dose escalation study. So that was exactly the process we did, and the highest dose in rabbits was safe, except for some local effects of cell injection. And then if you tried to do a spinal tap on a rabbit, you'd understand why. So here's the trial that's now ongoing, and what we're working on is that we have a patient, because of they have to have confirmed ALS, so they have to have a definite diagnosis, which means they're going to have significant disability. Now what we're dealing with is death of neurons in specific parts of the spinal cord and the brainstem. So the overall strategy that we're working on is we do an adipose tissue biopsy, like a mini liposuction. We take about 10 grams of adipose tissue. And then a critical part is that someone from the Human Cell Therapy Lab is there in the room and takes possession of that sample directly from the surgeon, who takes the sample, goes into the vial, and it's immediately in the custody of the Human Cell Therapy Lab, the GMP facility. We then take it through a standardized isolation procedure. Cells are grown up. At the moment, we're going with cells that are unmodified, because I'll talk very briefly about. The next part of this is to start to modify the cells as well in terms of being stimulating them to produce high quantities of neurotrophic factors. As you see, the first phase of the study is to use unmodified cells, then we'll move on to the modified cells. And then these will then go through a process of isolation of the neurotrophin secreting cells, grow those up in large quantities in the cell therapy lab. This whole process takes about 3 to 4 weeks. And so in order to have the right release criteria and everything, the patient comes into the clinical research unit 6 weeks after the fat biopsy, and the cells are then injected into the spinal fluid with a standard lumbar puncture. So when the cells go in, we know from the rabbit studies that the cells will engraft and line the meninges inside the spinal fluid compartment, and then hopefully will reduce the factors that will protect the neurons. So that's the overall strategy for this. So what we're looking at is in July 2010, we did a single patient as a pilot. We got an IND in July of 2012 for the dose escalation study. And this is the dose escalation that we're going through. So we're now a little over halfway through. We've got 14 patients enrolled, with 10 patients who've actually received their injections. And so far, everything's going well. After the injection, the patients in the clinical research unit for 3 days, for general systemic toxicity monitoring, and then they have repeat spinal fluid examinations 1 week and 3 weeks after the initial injection, and MRIs of the spine at 2 weeks, 4 weeks, and 3 months. Looking forward in the future, as I mentioned, the next phase, which has been through a pre-IND meeting already, is in association with the company Brainstorm, and this is to do the neurotrophin secreted modified MSCs. And that will be a 48 patient study that's shared between Mayo Clinic, Mass General, and University of Massachusetts. And then what we see coming out of this is selecting which cell types look most promising to move forward into phase 2B, much larger patient number trials. And potentially one of the things that we see in the future as well, the possibility of gene modifying cells to express specific neurotrophic factors. So this is a big process. This is our neuroregenerative and musculoskeletal regenerative team, and it's the cell therapy lab, Dr. Dietz and his colleagues, who really made this possible. So I'll stop there and